Okay, so it's really a great honor and pleasure to introduce one of our keynote speaker, Professor David Weitz from Harvard University in the U.S. And I want to tell you a little bit about Professor Weitz, and I think this is one of the rare examples of fine physics meets other disciplines. So Dave's career is really remarkable, and among the different topics that he studies, soft matter physics, biophysics, flow in porous media, microfluidics, and really made fundamental contributions to all of these topics in many exciting publications. I think that among the other things that I look and admire about his track record is, not a lot of people were able to do experiments, for example, in space, and Dave was one of those that have experiments conducted in microgravities in space shuttles, and also did contributions to things that we all use, like deodorants and other components that of our daily today use. He's a co-founder of 4 or 5 different companies. He's the editor on the editorial board of Proceedings of National Academy of Science and Nature Communication, and received many honors and prizes. He's a National Academy of Science member, and so on and so forth. One of the unbelievable recent activity that he was involved, that I found it to be extremely interesting, important, and fun, is that he was involved in making a new course in Harvard where they actually combined two great, my personal favorites, and this is combining physics and science with cooking. So that an additional aspect of one of his great additions to the fun of science and how it can be combined with our day-to-day activities. And without further note, I first want to give you a small contribute from us for you. Thank you for coming. This is for you.
Thank you.
And I'd like to welcome him to speak. And today he's going to speak about fluctuation motions in cells and how it can be probed with nanoparticles. And we met in a conference a week ago, and I heard some of those new ideas, and it's fascinating without imagination. So I'm sure that you're going to join it. The floor is yours.
Okay, so thanks a lot, Roy, for that blah blah introduction. I will try and live up to something. Actually, it's a pleasure to be here. It's a really beautiful location. I was telling Roy the last time I was here was before he was born. Okay, I'm going to tell you not about science of cooking, and not about all these other things, but rather something about some biophysics work that we do. The work is mainly that of a current student of mine, Ming Guo. So, in fact, in my group we have 2 Guos. They're both Chinese. Mira is also Mira Guo. She's not part of this work. She's American. She's Chinese heritage, but American. And she's also spectacular. So is Ming. So I call them my Guotu people. And Mira really understands what I mean, but I think Ming is slowly beginning to understand. He came to us from Beijing. One of my colleagues insisted that we accept him. And I was very dubious, and I was dubious about it for a while when we started, because he would never talk to me. He would never argue with me. And my rule in my group is if you want to be a member of my group, you have to argue with me. Of course, you graduate when you win your arguments. So he wouldn't even argue with me. But now whenever I see him, I know I'm going to see spectacular data, and he's really terrific. Cliff Brangwyn, a former student who is now a professor at Princeton, did a lot of the other work with Heitze Koinderink when she was a postdoc in the group. And we've benefited enormously from a long-standing collaboration from the theoretical side from Fred McIntosh, and a lot of the basic concepts come from him. We also have collaborations with people in BU. You'll see a little bit of work there. Extensive collaborations with Jeff Fredberg and Anwar Zhao, who are two people in the School of Public Health with whom we make a lot of measurements. And Jennifer Lippincott-Schwartz is somebody who, those of you who know something about cell biology will immediately recognize her. And she and I were at a meeting, a workshop for a while, and we started talking, and she doesn't know anything about physics, but I would probably claim I know nothing about biology by comparison. But she has this enormous intuition, and she made some suggestions which took Ming several years to sort out, but actually are turning out to be true. And this is based solely on her own intuition, which is really remarkable. And I'll show you what I mean as we go along. So here's an image that Cliff Brangwyn took of a cell. What he's done is he's transfected the tubulin so that it is fluorescently labeled with GFP. So all of these filaments that you see are microtubules. They've assembled, and you can see the structure within the cell of the microtubules. If you zoom in a bit, you notice that there are these granules, they're endogenous granules, these black things. You can see them here. I've just zoomed in on the image. And they are moving around in some kind of fluctuating motion. This is not a directed motion here. They're not being pulled by a molecular motor, but rather they're undergoing some random fluctuating motion. And coming from soft matter physics the way I do, when I see that, it reminds me very much of images like this. This is taken with a microscope. It's in fact a confocal microscope, but that's not important. It's a time evolution of the positions of two colloidal particles. These are probably, well, they're 2 micron diameter particles. This is slowed down slightly, and they're undergoing random fluctuating motion as well. But in this case, we understand very well how to analyze this kind of motion. It's Brownian motion, and we can analyze it in great detail. And if you look at this motion, your first instinct is to say it's also random fluctuating motion that has, for all the world, the character of Brownian motion. And it's an interesting, then, idea to explore to see how far we can take that analogy. So the thing about, or the important thing to recognize is that Brownian motion is intrinsically an equilibrium phenomenon. It's the equilibrium fluctuations due to thermal motion of a particle in a viscous fluid. But living systems such as the cell are inherently non-equilibrium because they're alive. And so we might ask, how far can we go by trying to take the tools of statistical mechanics, the tools of describing things as an equilibrium state, and apply it to the cell, or to the fluctuations in the cell, which is inherently a non-equilibrium system. How far can we do? What can we learn from this? And what I'd like to show you is that there are a lot of very intriguing analogies, times where you think it should work really well, but you will be really drastically fooled if you do that, and you will get things quite wrong if you do it, just as tempting as it is. So if we want to analyze Brownian motion, in fact, there was a beautiful poster looking at the same thing with Brownian motion near a wall with particles. The way we do it is really well understood. This is looking at the location of each of these particles. You can identify the center of a particle because you know its diffraction pattern. You can identify the center of an isolated particle to far greater precision than the wavelength of light. You can do it to a few tens of nanometers. You're not limited by the Rayleigh criterion for resolution in a microscope because you know the diffraction pattern. Okay, so this is just tracking the location of the particle, and this is the color is the function is time. You can see the position is as a function of time here. This is in one direction. And you can analyze these. You can calculate the mean square displacement. That's the standard way of analyzing diffusive motion. If you calculate the mean square displacement, it looks like this. It's linear in time. That's exactly what you expect for Brownian motion. You can go quite a bit farther because you have this enormous amount of information by tracking locations of particles. You can do second order analysis. This is the so-called Van Hove distribution. The Van Hove correlation, this is distribution of set size. This is not so important. It's just to say that you have a lot more statistics available. You can analyze these things in very simple ways. The fact that the mean square displacement is increasing linearly with time. The coefficient of increase is the diffusion coefficient, and it's the so-called Stokes-Einstein. Einstein really was the one to sort out the full description of that. You can describe the diffusion coefficient as this thermal energy term and this damping term, which is involves the viscosity of the fluid and the size of the particle. And so this, of course, leads to a very simple implementation of what's called micro-rheology, measuring something about the nature of the material by looking at fluctuating motion. If you know the particle size and you can measure the diffusion coefficient, you can find out what the viscosity is. So if we want to do the same thing to analyze the motion of particles in cells, we can analyze the motion of those granules, or we can do something perhaps a little more controlled. What we did was we injected fluorescently labeled particles. These are nanoparticles whose size varies between, say, 500 and 1,000 nanometers, or half a micron and a micron. And we very carefully coated the particles so that proteins wouldn't stick to them, to make them as inert as possible. And then we literally took a small needle and injected them into the middle of the cell. And then, of course, let the cell heal, let it recover a bit, and then measure the motion of the particles. So here is the same sort of tracks of the motion of these fluctuating particles. You can see they undergo the similar type of qualitative type of motion, qualitative type of fluctuating motion that we saw when we looked at the behavior of the Brownian particles. This is a very similar characteristic, very similar behavior. And we can analyze them in the same way. And if you look at this and calculate the mean square displacement, this is again plotting it on a log-log plot, exactly the same way as we analyze the behavior of these Brownian particles. This, for all the world, looks diffusive. It has a slope of 1. It really seems exactly like its Brownian motion. It looks like its diffusive motion. And of course we can take advantage. Now we're using really finely controlled particles. We know the size of the particles. We measure the diffusion coefficient. We can use the Stokes-Einstein equation, and we can calculate the viscosity. And you find that the viscosity that you get is something like 500 times that of water. That's maybe not so surprising if you ask what should it be inside of a cell, because everybody tells you the cell is a very crowded environment, and we know that crowding somehow reduces viscosity. So maybe this is not so unreasonable. But then you remind yourselves that, remember, I showed you one of the networks that was fluorescently labeled in those other cells. That was the microtubule network. In fact, there are several other biopolymer networks inside of cells that give cells. Okay, so how can we understand that? Well, what I showed you before, I was slightly lying. I was cutting the data off here. If I take the full set of data of these tracks and calculate the mean square displacement, you see that at longer times it is indeed diffusive, the way it should be for a Brownian particle in a viscous fluid. At shorter times, it undergoes a different type of motion. it seems to, there still is some fluctuations, but there's much less time dependence. In fact, here's a set of data. This was, those were just a few sets of data. This data, or these data, are an average over many, many different particles and many cells of the same sort. The one thing that's held constant is the size of the particles. Well, we can understand the nature of, we can understand the nature of this motion here, again, using some simple concepts of micro-rheology, that is, the idea of thinking of what thermal motion is in a more complex medium. And we can do this with a very simple conceptual idea. Imagine if I take a particle and I attach it to a spring. It still has thermal energy, but now it's attached to a harmonic spring. What's going to happen is it's going to fluctuate, and the energy that it has, the kT of energy, will be balanced by the stretching energy, the energy of the spring itself. So I can just balance the spring energy, which is proportional to the spring constant, and the fluctuation, the size of the fluctuation, well, that's what I'm measuring here, the size of the fluctuation, the square of the fluctuation. I can balance that, set that equal to kT, that's the energy that I have. So here's the energy, it's a half times the spring constant times the fluctuation squared, which is what I measure right here. I normally don't want to calculate a spring constant when I describe the behavior of a material. I'm much better off talking about an elastic constant. The difference between a spring constant and elastic modulus is one length scale. The only length scale here is the size of the particle. So by including the size of the particle, I can calculate the elastic modulus. And if I do that, I get something like a Pascal. Now, if you look in the literature of measurements of the elastic constants of cells, you'll see that it varies anywhere from a few pascals to thousands of pascals. In actual fact, the measurements of thousands of pascals are the outside, the outer side of the cytoskeleton of the cell. If you poke on the cell, or if you put some particle on top and twist the cell, that's where you measure something that's quite so large. If you put a particle on the inside, or if you measure the elasticity right in the center of the cell, it tends to be more like 10 pascals, so maybe 1 pascal is not unreasonable. So this suggests that perhaps we can describe all of the motion still by saying that it's thermal motion. It's some kind of viscoelastic property. It's elastic at some frequencies, and it's viscous at other frequencies. At least this conceptually or qualitatively seems to give some agreement or some description of the type of data that you see. Of course, if it really is micro-rheology, if it really is behaving as thermal motion, we can do simple tests. We can just vary the particle size, because all motion, all fluctuating motion, is the behavior on particle size is well known. It's basically inverse of the particle size, whether it's viscous, where the diffusion coefficient is inversely dependent on the particle size, or whether it's elastic, where it's also inversely dependent on the particle size. So here's a series of different particle size measurements, and we can just ask, do they scale together? And the answer is yes, they scale exactly as you would expect. So again, it's consistent with this simple-minded picture. However, as always, when you do an experiment, at some point you should stop and not do more experiments, but if you're careful, you always do a few more experiments. And so we did a few more experiments. In particular, what we did was we tried to inhibit some of the molecular motion. So to do that, we added blebistatin. What that does is it prevents some of the myosin motors, some of the molecular motors inside of the cell, from being active. And what you find, this is a little busy slide, but if you look carefully, you see these open points in each of the sets of data. You see that it changes the motion. It doesn't look like it's a significant change, but this is nearly an order of magnitude change on this log-log plot. It changes the motion. There is much less motion just by inhibiting the molecular motors. We haven't changed the temperature, we haven't changed the elasticity, and we haven't changed the viscosity. We've just inhibited the activity of one set of molecular motors. Now we can be more, take a large sledgehammer, be more decisive about how we inhibit motors, and starve the cell of ATP. It's the chemical on which all molecular motors operate. And if we do that, you see that now virtually all of this diffusive-like motion completely disappears. Again, we haven't changed the temperature. We haven't really changed the nature of the cell in any way. We've just inhibited the activity of the molecular motors in the cell. And the fact that the motion, the fluctuating motion, depends so sensitively on the molecular motors suggests that this simple-minded assumption that it's just thermal motion really doesn't work. So the temperature, the idea of using temperature isn't really a very good one. So how can we understand what's going on? In actual fact, absolutely not. The local temperature is absolutely fixed by the bath that it's in, which is essentially room temperature, 37, depending on how the cells were grown. But no, it doesn't change the temperature. And if you wait maybe 5-10 minutes, I'll tell you what temperature you would need if this were really temperature, because I have a better way of measuring, or a different way of measuring it, where I can actually determine what temperature is. Okay, and then you can ask me the same question and see whether it makes sense to talk about temperature. Okay, so how can we understand it? Well, right now we're making an assumption that we need temperature, so we know temperature, we're assuming temperature. If we want to actually measure an effective temperature or ask the question, can we separate the role of temperature, we need to have something whose properties, in particular its elastic properties, are known. So we turn to a different kind of measurement, and we look at some of the biopolymer networks themselves. In particular, we look at the microtubules. So here again is one of these cells which has been transfected with GFP-labeled tubulin so that you can see the microtubules, you can watch them. And now here is a time exposure. This is slowed down somewhat, but not that much. And if you look at them, you see that the microtubules themselves are undergoing fluctuating motion. In fact, if you remember the first image that I showed you, you also would see large fluctuations of the microtubules. Now, what's surprising about that is that the microtubules are themselves rather stiff elements, but we know their bending modulus. We know how stiff they are. So we can analyze these things and try and understand the nature of the motion. So the way you do that is that you look at the shape of the microtubules, and you project the shape into normal modes. We used cosine modes, and we looked at the angle-angle correlations. We looked at the distribution of width, of amplitudes of the angles of the microtubules, and we plot the amplitude as a function of the wave vector, as a function of the length scale that you look, and you see that you observe this very characteristic q to the minus 2 dependence, which is what you would expect for the angle-angle correlations if they were undergoing fluctuating motion. So again, this has the hallmarks of something that is undergoing thermally induced fluctuating motion. In fact, you can test this and sort out the nature of this by doing the same experiment now with a single microtubule. So what we've done here is we've taken the fluorescently labeled tubulin, we've reassembled a single microtubule in between two glass plates in a fluid, and we can look at the fluctuating motion. You can see that it's fluctuating. You don't expect to see enormous fluctuations because the microtubules are very, very rigid objects. But this is about 50 microns in length, and you can see there are some fluctuations. We can do the same kind of analysis, and when we do that, we see that indeed we get the q to the minus 2 dependence that you expect, but now it's about 2 orders of magnitude lower in the amplitude of the fluctuations. So the microtubules in the cell do indeed look like they are undergoing thermal fluctuations, but with a much, much larger amplitude. And since we know what the elastic or the bending constant of the microtubules are, we can actually now calculate what, if they were undergoing thermally induced fluctuations, we could calculate what the temperature is. This is your question. And the temperature we get is 30,000 K. So you might think that the cells are hot, but boy, they are very hot. And that seems perhaps a little unreasonable to think of that. Now, okay, let's step back, and I make fun of this, but we shouldn't, because what we're doing is we're trying to ask, can we use the concepts of equilibrium statistical mechanics, but just change the temperature to some arbitrary temperature? Does that make sense? And to a certain extent, it seems that it has some sense of validity, that you can see many things that behave like a thermal excitation, but with a much higher temperature. Now, in actual fact, if you worry about the details of that, this is really not such a good way of thinking about it, because now you should start thinking about the spectrum of fluctuations and whether you actually have the same spectrum that you have with a thermal fluctuations. So we should look more in detail as to try to understand the nature of this effective large temperature, or the nature of these fluctuations. Well, so if we go back to the microtubules, how can we understand something about the nature of their fluctuations? They're clearly undergoing these very large fluctuations that are occurring because of some very high effective temperature, if you will. And the clue to the behavior comes from doing exactly the same experiment that we did with the fluctuating particles, and that is we inhibit the activity of the molecular motors. And if we do that, then we completely lose any of the fluctuations. So again, the fluctuations are somehow driven by the molecular motors. But these are not necessarily molecular motors running along the microtubules, but rather the presence of all the molecular motors in the cell. So how can we understand the nature of that? Well, to do that, we turn to a reconstituted, an in vitro system. We reconstitute a minimalist model of what the nature of the cell might be. We do this, this is based on sort of years of work that we had done in trying to understand something about actin networks, but we can take advantage of our knowledge of that. What we can do is we can purify actin, the protein actin, which is the primary protein that makes up this network in the cytoskeleton. We can purify it and then reconstitute it to form a network, repolymerize it to form a network. And you can see we can make a network that has very similar characteristics, very similar morphology to the network you see in the cell. In order to do this, what we've done is we've added first a second protein called gelsilin, whose role it is to prevent the polymerization of the filaments from getting too long. So it keeps the filaments shorter. And second, we've added a third protein called filamin, which is a cross-linking protein to make the network. If we have long filaments, they'll entangle to make a network. But if we insist on keeping the filaments short, in order to make a gel-like network, we have to add a cross-linker. And so we do this. And then what we do is we add a model molecular motor. In fact, we add myosin, myosin 2, the same motors that you find in muscle. Those are one of the types of motors that we inhibited with the blebbistatin. And we add some labeled microtubules. We do exactly what we did in the glass slide. We repolymerize fluorescently labeled microtubulin to form a single microtubule. And then when we do that and we look at what happens, this is what you see. you can see that the microtubules are undergoing very, very large, relatively short length scale fluctuations in their shape. They're undergoing these large bends. Now, let me emphasize again that the molecular motors that we've added to the system walk along the actin network. They don't walk along the microtubules. So there are no molecular motors that are actually pulling the microtubules. Nevertheless, the microtubules are undergoing these very, very sharp bending fluctuations. See, this one's going to undergo one here. See how sharp it is. So how can we understand that? Well, here's a simple schematic model. The myosin motors that we use are these small filaments of myosin. They have about 300 myosin heads per filament, about 150 on each side. The duty cycle time of the myosin is about 2%. If you have 100 filaments, there's always one that's walking. So these motors are processive, and they can walk in one direction along a given actin filament, but they can walk, each head, each side can walk in one direction. So they have the effect of this head walking in this direction and this one walking in this direction, and that's like pulling on the mesh of this network. It creates essentially a forced dipole where it's pulling on one side and pulling on the other side. The network itself is, the filaments in the network are linked to the rest of the network. So if I pull on it, imagine me pulling on a fishing net. If I pull on it, it puts a force on it, it pulls a force on it. If now I have a microtubule that's embedded in here, if the network is being pulled here, the network will pull on the microtubule, and that will make it undergo this large fluctuation. So it's not the motors themselves, but rather this cross-linked network that's pulling on the microtubule and causing it to undergo these fluctuations. So we can do the same kind of analysis. If I go back to this image here, we can do the same kind of analysis of these correlations of the angles. And if we do that, we see something that looks like this. It raises the amplitude at the high short wavelengths, or the high wave vectors. It raises the amplitude rather considerably. Not quite as much as you would get in the cell, but still quite a large amount. So it raises the amplitude at the short wavelengths. That's what you expect because you saw these short wavelength fluctuations. So the question then is, why do you see, when you look at the angle-angle correlations, you see this q to the minus 2 over all wavelengths. In fact, you see also large fluctuations at very short wave vectors or very large wavelengths. Where do they come from? Well, a clue to that comes from looking at not the ensemble of all structures. So the way we did the analysis in the first place is we took a lot of different images and we measured, on each image we measured the angle-angle correlation, and we assumed that by taking lots of different images we were getting an ensemble average. But now if we look at just a given set of microtubules and look at the temporal behavior, just measure the time-to-time difference, these are images where what we've done is we've taken two sequential images and subtracted them so we can see just the difference. And so the red indicates those parts of the microtubules that are a new position, and the green is their old position. So that represents the change. And of course, what you see is that all the change occurs at high wave vectors or short wavelengths. It doesn't occur at these long wavelengths. Nevertheless, there are fluctuations, there are shape fluctuations at long wavelengths. Where do they come from? Well, a clue comes from recognizing that the microtubules themselves are always growing. They grow for a while, and then they spontaneously depolymerize and collapse all the way back, and then they grow again, and they collapse all the way back. And this is a well-known behavior of microtubules that I think Tim Mitchison first discovered 20-odd years ago. And so you can watch how a single microtubule, what happens when it grows. And Cliff caught a microtubule, and he's highlighted it in red as it grows. So this is just a sequence of images over time, and you can see it's growing, and then spontaneously it's subjected to a transverse fluctuation that's very reminiscent of what we saw in these in vitro networks. It gets this large length scale, large radius of curvature bend, but now it grows, but it's growing in an elastic medium because it's in an elastic network. It's embedded in this actin network or whatever the network is underneath. So it's growing in an elastic medium, so when it grows, it can't relax back. The bend is fixed into place because it's growing through this elastic medium. So as it grows, and you now have a very large length scale curvature. So the large length scale curvature are more permanent. They come from the transverse fluctuations that occur during the growth of the microtubules. Actually here, to show that, you can fluorescently label a tip of the microtubule so you can just see the growing part of a microtubule, and you can analyze its shape. And if you do that, you find that just looking at the shape of the tip, as it grows, gives you exactly the same angle-angle correlation, that's q to the minus 2 behavior, that you get for the large length scale for all the fluctuations of the microtubule. So there's a combination of these dynamic fluctuations due to the motion at short wavelengths and the consequence of these dynamic fluctuations due to the fact that you're imposing transverse fluctuations on the microtubule as it's growing. So you can analyze this in more detail. Here is the shape of one of these microtubules in this reconstituted system. And what we've done is we've plotted the location. You can identify the location the same way you can identify the location of a particle. You can identify the location of the microtubule. And we fit it to a solution of a rigid rod that's subjected to a point force. This is a homework problem in Landau and Lifshitz. So we solved that. The referee or the editor of the paper of the journal that we published it in, which is a very biological journal, said he'd never published a paper in the biology journal that referenced Landau and Lifshitz's mechanics book before. So we were happy about that. But you can fit this. You can get the elasticity or the elastic constant, the bending constant of the microtubule. It's exactly what you expect. You also can get the amplitude of the force, and you find that it's about 5 to 10 piconewtons, exactly the kind of force that molecular motors can exert. So this is all consistent. Now the other thing you can do is look at the temporal behavior in the cell. And this is very important because it begins to give you a clue as to what these molecular motors can do. If you plot the amplitude of this fluctuation as a function of time, this is just some time dependence, what you find is that it goes up fairly rapidly, and then it stalls. It stays in place for some period of time, and then it lets go and relaxes completely. This is exactly what you would expect for a motor, a processive motor. It will pull for a while until it reaches the force at which it stalls. Then it pulls, it pulls, it pulls. It can't let go. It can't go farther because it's reached its stall force, and then it lets go. And you see something that looks like this. So this gives you a characteristic frequency or a time evolution of the forces of the molecular motor. And once you know you can think of this as a fluctuating force due to the molecular motors. And this is a direct measurement of what they are. So this, if you look at the frequency dependence, you can analyze the frequency dependence of motion like this. And Alex Levine and Fred McIntosh did that. And what they showed is that gives you omega to the minus 2 dependence. That turns out to be very reminiscent of what you would see for Brownian motion, but it's not that the forces, it's not that the medium is exhibiting this kind of fluctuation, but rather the forces. There's a spectrum of forces that are exhibiting this kind of motion. So now we can go back and ask, let's imagine that we have a particle, this fluctuating particle, embedded not in a viscous medium, but rather a network, which is more typical of what we might think, what we would think. But now we imagine that we have a random distribution surrounding this particle of molecular motors. Each molecular motor is like a forced dipole. So you have random forced dipoles that obey this particular frequency dependence that I showed you, that it pulls for a while and then lets go. So it goes up quickly, it's flat, and it lets go. And you can Fourier transform that to get the kind of response that you would get. And what you find in this case is the motion of the particle looks for all the world like it's Brownian. It increases nearly linearly with time. And this is, again, something that Fred McIntosh and Alex Levine pointed out originally. So if that's the case, if this is correct, let's do some kind of test to verify that. We should, first of all, find that, in fact, the medium inside the cell is not viscous, like it seems to be if we interpret it as Brownian motion, but instead is really truly elastic. So to do that, rather than depending on the fluctuating motion, the random motion of the particles, what we did was we imposed a motion. So we worked with Jeff Moore's group and Mikkel Jensen, his graduate student, who very conveniently for us turns out to be the husband of one of our students. And Ming took his cells over to their really beautiful laser tweezer setup, injected particles, and then pulled on it with an oscillating laser tweezer. This is a very difficult measurement to do, but they were able to do this. These are the results. This is the motion, the oscillating motion of the drive of the laser trap. So we're pulling the laser trap, we're oscillating the laser trap, and we're measuring the resultant position of the particle. And the important thing to look or to notice here is that the position and the drive are very nearly in phase. If this were a fluid, it would be exactly out of phase. If this is a solid, it's exactly in phase. The fact that this is very nearly in phase means that this is truly a solid. And we can vary the frequency. We can measure both the amplitude and the phase difference. That allows us to measure the real and the imaginary part, the viscous and the elastic part of the response. These are shown here. This is the elastic part. This is the viscous part. The elastic part is larger than the viscous part. In fact, this kind of characteristic, this is measuring the elastic modulus, the viscous modulus as a function of frequency. This is very reminiscent of what you see for any soft elastic material. You can, if you want to describe the elastic part, you'll say that it's nearly independent of frequency, but not quite. In fact, its frequency dependence is about 0.1. So now we can go back. We can, this sort of confirms the fact that the material inside the cell is indeed elastic. We can't go to the lowest frequencies that we need to see all the way down here, but if we reinterpret the data that of the elasticity in terms of a mean square displacement, you see something that looks like this. And there's no evidence that it's changing here, so we're very convinced that the material is elastic even at these long time scales. So it's clear that we're seeing an elastic material, and that the motion then is much more the result of the fluctuations of this elastic network due to the random location or the random positions of these motors, the forced dipoles. We can actually go quite a bit farther. What I showed you, all the data that I showed you were ensemble averages over many different particles. If we look at the mean square displacement of individual particles, we notice several different things. First of all, we notice that when there's a power law increase, it's generally somewhat larger than 1. If we average this at longer times, then they all seem to begin to flatten out. What we've done is we've averaged these where it's a larger increase than 1, but then average with these particles that are flattening out. The sum, the average of all of them gave something that looked a little bit closer to 1, the slope here. But in fact, the slope is always a little bit larger than 1. This is actually quite significant because if you look at the analysis of the slope that you would expect, you would expect, given that you have these random force dipoles with this particular frequency dependence, you expect from that this linear dependence. But because the medium itself has a frequency dependence, remember it was 0.1, you also have to incorporate that dependence, and the way you do it is the slope that you expect should be the 1 due to the frequency dependence of the motors, and 2 times the slope that you have. So that would be 1.2. So this slope should be closer to 1.2, and if you actually look here, we're just plotting the instantaneous slopes, and you see where it's power law, it is indeed much closer to 1.2. In fact, here's a distribution, and it really does peak somewhere around 1.2. So this is consistent then not only with the nature of the fluctuation motion, but also with the additional consequences of the elasticity of the medium. You can go a little bit farther. You can actually calculate. Remember, I showed you that the mean square displacement started to saturate. That's due to the fact that the motors eventually stop operating. And so by looking at the distribution of times, you can get something like a processivity time. You can get how long the motors are actually processive. This is something that is well thought of, and you can see with single molecules, but it's very difficult to have this kind of measurement of a random distribution in cells. Here's a way we can do it somewhat directly. The other thing we can do, and I won't go through the details of this, I'm happy to talk about it later, is we actually can calculate the spectrum of forces. So now what I'm saying is that we have, rather than having a flat distribution of forces the way you do for thermal energy, we have a omega to the -2 distribution of forces because of the nature of these, of the processivity. Basically, it's because of the Fourier transform of this decay. And here we see that this data is actually a collection from another study that we were trying to do to understand the nature of changing different parts of the cell, and we can see that by just changing the nature of the cell, we can see whether the nature of the spectrum of forces that you get in these fluctuating motors, whether it changes under different conditions. Okay, so this suggests that the fluctuating motion is not thermal, but rather it's induced by these molecular motors. We can do a few other things, and that leads to some other very interesting, intriguing results. This is incorrect, but I want to somehow parameterize the amount of motion that I have so I can compare it for different regions. So I can sort of use concepts, again, from statistical physics, from what I would do from analyzing other types of motion, and say that the motion here is somewhat like diffusion motion. Let me define some kind of diffusivity. I can do that, for example, by asking how far does a given particle move at a certain time. That gives me something like a diffusion coefficient or something like a diffusivity. I do that only to parameterize behavior, because if I ask how does it depend on different parameters, I see, for example, that if I shrink the volume of the cell, I do that by applying an osmotic pressure on the outside. I put PEG on the outside, and I shrink the volume. I squeeze out the water. Then the diffusivity decreases. And in fact, you can see that it sort of decreases. This is just different particles. It's scaling with different, or it scales with particle size, sort of the way you would expect. But that suggests that volume is also really an important quantity. So let's go and look at the volume of cells and try to understand something about the nature of the volume of the cells. Well, you can change the volume of the cells, it turns out, in a number of ways. The standard or simple way of doing it is to squeeze water out using an osmotic pressure. But here's another way that you can actually do that. If you look at a cell, this is a sort of a well-known effect. If you look at a cell grown on substrates of different stiffness, you see that the cell spreads to a different degree. It tends to like to adhere to a very stiff substrate. This is a stiff substrate. And the cell spreads much more when it's grown on a stiff substrate. This is glass, and this is a rather weak gel. And so, in fact, you can plot the area of the cell as a function of the substrate stiffness. This is for different classes of cells. And you see that for very soft substrates, there's not much change, but then it increases rather significantly. And you can see this in these images, so that when it's plastic or glass, some very rigid substrate, it's really spread to a much larger extent. Well, this is actually well known, but perhaps what's not quite as well appreciated is as it spreads, its height also decreases. So here are some confocal images. This is the XY plane, and this is the XZ plane. And you can see that as it spreads, it actually gets lower. The average height decreases. And so here's a plot now of the height of the cell. So very roughly speaking, the product of these is the volume. In actual fact, we measure the volume rather precisely just by using a confocal microscope, dyeing the cell and measuring precisely the volume. And if you look at the volume of the cell as a function of the stiffness of the substrate, you see that there's a very sharp decrease in the volume. So the cell spreads out, but it pulls itself down and actually has to expel water due to the fact that it's on the stiffer substrate. Okay, this is just to check that these are, we did this average over many cells, so we believe these results. You can see a lot of other interesting effects. I won't go into this in great detail, but here is a cell where if we put it on glass and we inhibit the motors, then it can't spread and the volume can't decrease. If we grow it on a substrate that we can cross-link by exposing just briefly to UV, then we see that it starts at a much larger volume. We make the substrate thicker, and after say an hour to equilibrate, the cell volume decreases. So it really doesn't depend exactly on the substrate that you grow it on. It depends really on the stiffness of the substrate. But then we can do other experiments. We can take it, so it seems that it depends on the stiffness of the substrate, but what we can do is we can do micro-contact printing. The cell is adhered to the glass, not actually the glass itself, but the glass is always coated with something that the cell likes to adhere to, fibronectin or collagen. We generally use collagen. And we can micro-contact print the collagen in defined regions. So here we're micro-contacting printing them, so we're controlling the area of the cell on the stiff substrate. So this is glass, but we're controlling the area. We're not letting it spread by preventing it from adhering to a larger region. And if we do that, we also see that the height changes. And in fact, if we then look at the cell volume as a function of area, this is all on a stiff substrate. We see exactly this dependence. So it doesn't seem to depend on substrate stiffness, but really it seems to depend on adhered area. And you can do the same thing by, this one is on a gel. So the substrate is retained stiff, and we're shrinking the area, we're changing the area with the substrate by changing the substrate. And again, the cell volume seems to correlate with the area. Now, why is this interesting? Well, it's interesting because what's normally thought, if you go back in the literature, you see that there's a very common measurement made by many different groups where they now measure not the volume of the cell, but rather the stiffness of the cell. And it's well known that the stiffness of the cell correlates with the stiffness of the substrate. Here's the stiffness of the cell, substrate stiffness. This is fairly widely observed. It has a lot of very strong implications. There's some beautiful measurements that suggest that stem cells differentiate differently. They go to bone if they're on a stiff substrate. They go to neurons if they're on a soft substrate. So the substrate stiffness is a cue for the differentiation. So this is a well-known effect. However, now if we go back and measure the stiffness of the cell. So here's the stiffness of the cell. And what we did was we control the volume. This is on a soft substrate where the volume is large. We control the volume. We decrease the volume by using an osmotic pressure, and we find the stiffness increases when we decrease the volume. Similarly, if we go on a stiff substrate, so it's on a stiff substrate where the stiffness of the cell should remain stiff, if we do that and we control the volume by controlling the area, we find that again the stiffness also decreases as we increase the volume, as we confine the area. So there is no correlation, no direct correlation between cell stiffness and substrate stiffness. Instead, there's a correlation with volume. Let me skip this and just show you. So here's measuring stiffness as a function of cell volume. Here we're changing the substrate stiffness. We change the adhesion pattern. We change the osmotic pressure on a soft substrate. We change the osmotic pressure on a stiff substrate. We use a patterned substrate. We can vary the volume in a whole bunch of different ways, and we always see this correlation of the stiffness with volume, and in fact, if we plot all the data together, they scale very precisely. So it seems that cell volume is a better measure of cell stiffness than is substrate stiffness. So this calls into question all these measurements that suggest that the Q for stem cell differentiation is simply the substrate stiffness. One other thing that we can do, which is, again, very intriguing along these lines, is we can measure the volume of the nucleus. We can label the nucleus, and so you can see here are different cells grown in different conditions. And indeed, the nucleus volume also tracks the cell volume. So as you decrease the volume of the cell, you also decrease the volume of the nucleus. Now, this is particularly important because nuclear volume is essential for the transcription of proteins inside the cell. And this is one of the cues, one of the important things in differentiation. So this could also be a very simple reason why you get this different differentiation, but it seems that it's volume and not cell stiffness. Okay, so this is, that part is very much a work in progress. We're trying to sort out really what the meaning of the cell volume is, and we're of course trying to ask the additional question, as the nucleus volume changes with the cell volume, does that change gene expression and other important attributes of the cell? The indication so far is that it really does have a very strong effect on that as well. Okay, so I hope I've convinced you that first, fluctuations in cell motion are not a function of temperature, but rather a function of the motion, and the function of the motion of everything inside of the cell depends very, very critically on the volume of the cell, and that seems to have tremendous control over a lot of properties of the cell. So with that, let me take any questions you have.
Some of the differences, important differences. Is there here also a parameter like temperature, which not only that you can define, but that you can control in a continuous way? I mean, you show that when you inhibit motors, you have very different behavior, but can you somehow tune something?
So I think the answer is not easily, but if you think about it, the tuning parameter that you would have, remember I showed you the force spectrum. The amplitude of the force spectrum would be the equivalent of controlling temperature, and that would be controlling the activity of the motors. So if you could somehow controllably vary the activity or the amount of motors, then I think you could achieve that degree of control.
You mean the amount of motors, not each of them particularly, but
I think the amount. I think just the number, because it's the amplitude of the spectrum.
I'm very happy seeing your last set of data because completely agrees with our measurements. And I can show you through data nothing showing also that material stiffness does not regulate stem cell differentiation. The second part, since you initially started out with Brownian motion and then mentioned the role of motors in mediating the fluctuations. Did you also work at Chaoul, which rigidifies the microtubules and slows down the depolymerization rate? And that would be an experiment that could be done ex vivo and in vivo to just compare how the rigidity of the microtubules impacts all the phenomena that you've seen so far.
Okay, two things. First of all, the differentiation of stem cells with substrate stiffness, there is a beautiful paper that shows that, and its most important attributes are, first of all, everybody knows about it, and secondly, nobody can reproduce it. But it's still a nice paper. In terms of Chaoul, I don't think we've used Chaoul. We've used a lot of other pharmacological agents to depolymerize actin, to depolymerize microtubules. I don't remember us using Chaoul. We used, we've gone the other route of depolymerizing rather than stiffening. It's a good idea.
Since cell volume certainly changed by applying pressure, and we know that even animals like us can survive at much higher pressures than the normal pressure, has anyone tried the experiment of letting a pregnancy of some animal develop not under usual pressure, but under much higher pressure, and see if that changes the differentiation of stem cells?
Okay, so really what you're asking is if you change the volume of the cells, you could do that by pressure, by any other means that you can do to change the volume, would you change the differentiation? The answer is, of course, I don't know. There are no examples that I know about that. I think what we're working very hard to test is whether we can see changes in differentiation of stem cells as a function of pressure, so individual cells. So that's more along the lines of just doing the same experiment. And all the indications so far, and this is not something I will dare show, but all the indications so far is that it does. So if you could do that under pressure, some people might say, well, they're always under pressure, and so things should be small, but that's not quite right.
Do it under 5 atmospheres, for example.
Well, it's not hydrostatic pressure, it's osmotic pressure. Okay? That's important. You don't want to squeeze everything because you can't compress water. Right? So when I say pressure, what I mean is osmotic pressure, I squeeze water out. So it's different than hydrostatic pressure. So you can't just put in a pressure chamber. You've got to put in salt.
What if the particles are smaller than the average size of the mesh, and is it biologically significant?
The answer is that's exceptionally important because, so let me tell you what this all means. Okay. First of all, the answer is that one of Jennifer's postdocs has done experiments with just GFP, and he sees no evidence of this behavior. It's just Brownian. So at very short length scales and very short time scales, Brownian motion probably dominates. At longer time scales and longer length scales, then you see this other effect. Now, why that's important is that where does fluctuations, where do they have to play a role? They have to play a role in any kind of chemical reaction because particularly if things have to identify or you have proteins have to find exactly the right location, that's probably always going to be driven by Brownian motion. But motion on larger length scales, that's going to be driven by this sort of stirring of everything. So here's an interesting observation. If you look at the size of cells that don't have molecular motors, they're always much, much smaller than the size of cells that do have molecular motors. And a simple explanation is that the effect of the molecular motors, not the motion of the motors, the pulling of the motors, but just the stirring of the motors, will increase the effective diffusivity of nutrients of everything so that they can spread over much larger volumes in some reasonable time than in the absence of that where you're strictly limited to Brownian diffusion. So there's a very simple explanation why cells with molecular motors are always much larger than cells without.
So
So if I understood you correctly, the microtubule flotation is mainly driving by myosin 2, and you didn't, or did, I mean...
So you didn't understand me correctly because I didn't say it clearly. In the in vitro system, the reconstitutive system, it was absolutely driven by myosin 2. In the cells, it's probably driven by a lot of different fluctuating motors, and I wouldn't know all of them.
Okay, so the microtubule motor, the diamond kinesine, basically generating force on opposite direction. So do you think that there's some sort of regulations on numbers of motors that can create some sort of directionality of the fluctuation of the microtubules leading to a directional movement of those particles?
So I think that motors themselves are incredibly well regulated. But what I'm showing is if I take the fact that they're regulated randomly or differently throughout the cell, and the net result, if I sit right in the middle, is that I see this random motion of all these motors, each of which is regulated on its own, that's what gives this the fluctuating motion. I think that for motion of molecular motors on any given application of the motors, any given use of the motors, they're probably incredibly well regulated.
So I want to talk about cell-to-cell variability and how does it sort of correlate, let's say, changes in cell-to-cell variability in volume, how does it correlate to cell-to-cell stiffness, and how much can you explain some of the changes in cell-to-cell stiffness by cell-to-cell volume?
I mean, that's a really important and good question. And I know that what we've done is look at the distribution of each of the measurements themselves. And every time I make a plot, there's a point and there's error bars, which are the variance of the distribution. And we've made measurements over 200 cells or so or more for each point. But I don't know if we've gone back and tried to ask, can we correlate within a distribution? Can we correlate? We have the data. I don't know. Well, I think we have the data. I'm not sure. Because measuring the stiffness is a different measurement. So that's a good question. I'm not sure we have the data. It's an interesting question. All I can do is ask Ming. I don't know.
I have a small question. What do we know inside the nucleus itself? So do we know anything about the fluctuations inside of the complex of the DNA, histones, whether that's going to be... Because if the nucleus volume is changing and that affects the relative motions of expression, then that might be also very important because it's a very light environment.
Yeah, so that's a great question. And as far as I know, you're asking something that right now people are starting to address. So I tell you two measurements that I know about. One was recently published by, I think it's Julie Theriault, who measured fluctuations inside of, I think she measured inside of, I'm going to say yeast nuclei, but I'm not sure. But she measured motion of something, she tracked something, I forget exactly what it was, and she saw some motion that depended on ATP. Okay? At the same time, there's a postdoc who works with Tim Mitchison and sort of between his lab and mine right now, Alexander Zidowska, who was from Cirrus's lab, maybe you know, who's done some really beautiful measurements looking at correlations of motion in the nucleus. And there's incredible amount of correlations over very large length scales, very long time scales. She does that by labeling histones and tracking or doing essentially PIV on the histones. So there's enormous amount of correlation, but that's just something she's really trying to sort out right now exactly what it means. The implications are to me, the implications are exactly what you think. That is that the transcription requires some unwinding of the DNA, and that takes space. And there are effectively molecular motors of the other ribosomes that are doing all this transcribing. So that is acting like molecular motors. So I think there's still a lot that's going on in the nucleus that's strongly affected by both the volume and whatever activity is going on. But exactly how they are, I don't know yet.
Okay, so let's thank Dave again.